Hello everyone, this is a review of genetics, in particular chapter 6 and 7 for the SBI3U course, grade 11 University Biology. I'm going to start with the first note, origins of genetics. Hereditary information is passed from generation to generation in distinct packets called genes. It is possible that some genes may not be expressed for many generations, but will eventually be expressed in future generations, lay silent or dormant, so to speak. In the 1800s, there were numerous attempts to explain how inheritance works, mainly with plants. It was morally more acceptable at the time to use plants. Few came to realize that plants can technically sexually reproduce. In the middle of the 19th century, a monk named Gregor Mendel did many experiments to explain the basis of heredity. He performed thousands of experiments by crossing or mating mice, bees, and several plants, but mostly his work was on the common pea plant. Pea plants were chosen because they are easy to grow, self-pollinate, and exhibit many distinct, obvious, contrasting traits. Here are the traits he investigated. The characters he studied, dominant traits and recessive traits, are what I'm going to be talking about. First character he studied was stem length. The dominant trait was tall, recessive was dwarf. Seed shape, smooth is dominant, wrinkled is recessive. The third character studied is seed color. The dominant trait was yellow, recessive was green. The fourth character studied was the pod shape. Dominant was inflated, recessive was wrinkled. The fifth character studied was the pod color. Dominant trait was green, recessive trait yellow. The sixth character studied was the flower color. Dominant was purple, recessive was white. And the final character studied was the flower position. Dominant trait was on the stem, recessive trait was at the tip. Next, we are going to be discussing Mendel's experiments. Mendel identified 7 pairs of contrasting traits. Observable characteristics or traits are called phenotypes. Mendel spent several years self-pollinating the pea plants in order to establish pure lines, known as purebred plants. Tall plants, for example, were repeatedly self-fertilized for many generations to ensure that all plants in future generations would all be tall. Mendel understood the value of having controls in his experiments. For this reason, he began by studying crosses between pure breeding plants that were different for only one contrasting pair of traits. These parent plants were called the parent generation, or the pea generation. When crossing a pure tall plant with a pure dwarf one, all the resulting plants were called hybrids. All the hybrids in this first filial offspring generation, called the F1 generation, were all tall. There was not one dwarf plant in the F1 generation. The trait that was expressed, tall plants, dominated over dwarf traits. He called these overpowering traits dominant traits. The trait that was not expressed in the F1 he called the recessive trait. Next, Mendel crossed his F1 generation plants with each other. For example, he was crossing capital T lowercase t plants with other capital T lowercase plants. He wished to determine if the F1 tall plants were identical to the P generation tall plants. Mendel found that after crossing F1 plants, the resulting plants, called the F2 generation, yielded about 3 tall plants for each dwarf plant, the mathematical ratio of which was 3:1. Mendel's experiments: F1 generation, the offspring from the P generation. The F2 generation is the offspring from the F1 generation. Mendel crossed one short with one tall pea plant. All of the F1s were tall, and 3/4 of the F2 were tall. He reasoned from this that the F1 generation was somewhat different from the P generation. He carefully repeated this experiment for each of the other 6 pairs of contrasting traits. All results were similar. The F2 generation displayed a phenotypic ratio very close to 3:1, where 3 showed the dominant trait for every 1 recessive trait in the pair. That's on that previous chart that I talked about, the stem length, seed shape, seed color, pod shape, pod color, flower position, and the dominant and recessive traits that go with them. Next, we're going to be talking about Mendel's law of segregation. So Mendel noticed a pattern in his results, and he suggested that there were units of inheritance responsible for this. For a given characteristic, there were several forms of these genes. These different forms are called alleles. Alleles are very important. For example, the gene for plant height has 2 different alleles, a dominant tall allele and a recessive dwarf allele. Mendel concluded that each plant's phenotype was determined by a pair of alleles that could be identical or different. Genes are located on chromosomes, a pair of homologous chromosomes. And you'll see on the picture a gene locus, or the address of a particular gene on a chromosome. The 2 members of a homologous pair carry alleles for the same genes, and therefore they affect the same traits. Mendel's law of segregation. Members of a pair of alleles for a given trait are segregated, separated, when gametes are formed. Mendel also saw that one allele from the pair had the ability to express itself dominant, while the second one is not expressed. It is recessive. There had to be 2 recessive alleles for that trait to be expressed. Phenotype is what the organism looks like. Genotype is the actual gene makeup of the organism. For example, using the letters TT, SS, 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 like in the picture. But the phenotype is just what it looks like. It's green and smooth, green and smooth, green and wrinkled. Mendel used capital letters to represent dominant alleles and lowercase letters for recessive genes. For example, if a pea plant was tall, it was either capital T, or capital T, lowercase t. As long as it had one capital T, the dominant gene, it would be tall. To be short, it has to be 2 lowercase t's, because it has to have both recessive genes. If the alleles for a trait are identical, for example, 2 capital T's or 2 lowercase t's, it is called homozygous for that trait. If the alleles for a trait are different, for example, capital T, lowercase t, it is heterozygous for that trait. Think about couples. Like if you're homosexual, it's only males, TT or lowercase t, or 2 lowercase t's, like say the capital T's are gays and the lowercase t's are lesbians, and the heterozygous is like a heterosexual couple. There is a man and a female, a capital T, a lowercase t. Recall that Mendel took a pure line of capital T plants, homozygous tall, and crossed them with lowercase t plants, homozygous short. This is the P generation cross. The F1 generation that resulted were 100% capital T, lowercase t plants, heterozygous tall. The genotype was heterozygous tall, and the phenotype was tall, because they still appeared tall even though they carry that recessive allele with them. Now what he did is he crossed the F1 plants with the F1 plants. When 2 heterozygous F1 plants are crossed, it is called a monohybrid cross. One trait is crossed. This produces different results from the first parental cross. Here, each plant can produce 2 types of gametes, T or a lowercase t allele. The resulting F2 generation, 75% tall plants and 25% short plants. The tall plants consisted of 25% capital T and 50% heterozygous capital T, lowercase t. The short plants were 25% lowercase t. Just think about it like on a Punnett square. When you draw it out, those are the results that you get. Then, of course, you'll see the picture there. You have the 2 parents, the tall, the short. Then the F1 turns out all tall. F1 mates, and then they get 1, 2, 3 tall, 1 short, like we talked about, that 3-to-1 ratio. Next thing we're going to talk about is Mendel's Law of Independent Assortment. Mendel then turned his attention to what would happen if more than one pair of characteristics were used in a cross. He began with a P generation cross involving a dwarf plant with purebred round steeds. That's TT, two lowercase t's, 2 capital R's. And a purebred tall plant with wrinkled seeds. Capital T, 2 lowercase r's. As a result of this experiment, he developed his second major law, the Law of Independent Assortment. When 2 or more pairs of characteristics are considered at one time, each pair shows dominance and segregation independently of the other. So, what I'm saying is the lowercase t, capital R, the capital R, and the capital T, 2 lowercase r's, they don't affect each other. The T doesn't affect the R doesn't affect the T. After this P generation cross, each parent produces only one type of gamete. They only give away one from each. The first plant produces gametes with a genotype of lowercase t, capital R. Remember, it was lowercase t, capital R. So the genotype that it gives away is it has to give away one lowercase t, one capital R. And the second plant only produces gametes with genotype of capital T, lowercase r. Remember, it was capital T, lowercase r. The only option is for it to give away a capital T and a lowercase r. And you'll see that when you look at the F1 cross picture on the side, you'll see which ones it can possibly give away. So the only possible genotype in the F1 generation is capital T, capital R, lowercase r, a dihybrid. All F1 generation plants are tall with round seeds. But when Mendel then crossbred 2 of these F1 plants in what is termed a dihybrid cross, capital T, capital R, lowercase r, times capital T, capital R, lowercase r. The Law of Independent Assortment would allow each parent plant to produce 4 different gamete combinations, capital T, capital R, capital T, lowercase t, capital R, and lowercase t and lowercase r. When these 2 F1 plants were crossed, 16 4x4 grid combinations had to be considered. A classic ratio of F2 phenotypes of 9:3:3:1 is found. Of course, that's 16, and our 4x4 grid that you can see on the side here. You can see all the different combinations. The next thing is genetic analysis. Punnett squares. These are extremely important. Punnett squares illustrate the possible outcomes, offspring of a particular cross. The possible genotypes of the gametes of each parent are placed on the exterior of the square as indicated. Consider the cross capital T, lowercase t times capital T times lowercase t. Consider the cross capital T times Consider the cross capital T, lowercase t times capital T, lowercase t. 2 types of gametes are possible from each parent, capital T or lowercase t. So each parent can give away a T or a T by chance. So if you set up the square, of course you have your capital T, your lowercase t, your capital T, your lowercase t, and then you put them together like a little multiplication thing you learned in grade 6. First square, you have TT, just makes sense. Next square, you got heterozygous, TT. Next square, TT, heterozygous again. And then homozygous again, you got homozygous short, lowercase t. Observe that the following genotypic ratio is capital T, lowercase t, lowercase t equals 1:2:1. So 25% are short plants. This is the classic 3:1 ratio of tall to short plants, if you're looking at phenotypic ratios, right? Another example of a Punnett square, but for a dihybrid cross. Cross capital T, capital R, lowercase r, with capital T, capital R, lowercase r. 4 possible gametes are made from each parent. So there is now a 4x4 grid with 16 possible outcomes. This is the classic 9:3:3:1 ratio that results. Look at the tables here. You have tall round, tall wrinkled, short round, short wrinkled. You get that same 9:3 to 3:1 phenotypic ratio. Same thing on the next one here. The chromosomal basis of heredity. We now know that one of each of the pairs of factors, genes, determining a trait in the pea plant is located on each chromosome of a homologous pair. Each gene is found at a specific place or locus on the chromosome. I'll repeat that. Each gene is found at a specific place or locus on the chromosome. If a pea plant with a capital T lowercase t genotype produces 400 gametes, meiosis would produce 200 with the T allele and 200 with the lowercase t allele. Test cross. A test cross is the cross of an individual whose genotype is being tested to a recessive individual. The recessive individual must be homozygous and so can only produce one type of gamete. So you can look at this little table and you see how a test cross would work, and that would be beneficial because you can find out, if you have the recessive trait or if you carry it and what the situation is. So next thing we're going to talk about is genetics after Mendel. Incomplete dominance. Mendel, after using the concept of dominance to explain his results, saw instances in nature where strict dominance didn't apply. He noticed that in some species or certain traits, intermediate phenotypes were observed. For example, when Mendel crossed red flowers with white flowers, both homozygous, he would get pink flowers. Neither gene is completely dominant over the other when it's heterozygous, but by crossing the pink F1 generation together, the F2 showed red, white, and pink. They came together. None was dominant over the other. They just kind of mixed together, and then they made that combo color. In humans, many traits show incomplete dominance, like most hair, skin, and eye colors. Think about it. Black person marries a white person, they have a kid, it pops out mixed. It's pretty simple. Then we have codominance. This is where two alleles are expressed at the same time. For example, in livestock, an animal can be homozygous red or homozygous white. So, you don't have a kid who's, a mix of the two. You have a kid with white patches and black patches. So when the offspring gets one allele from each parent, it expresses both red and white hair throughout the animal. The new color is called roan. Human blood types in the ABO blood group system show codominance. The possible blood types in this system are A, B, AB, and O. Your blood type is determined by a pair of alleles. However, in this case, there are 3 different alleles that may be found at the locus on either of the homologous chromosomes. When there are more than 2 alleles possible for a given gene, the condition is termed multiple allelism. In human blood types, the alleles for A, represented by capital I and then a little a up in the top, and B, represented by a capital I and a little lowercase b, are both codominant over the allele for O, represented by a lowercase I. Neither blood type A or blood type B can dominate the other. If a person has the genotype AB blood, then the phenotype is blood type AB. So of course, in summary, you can look at this genotype table. You can see the A blood or the A blood with a carrier of the O blood. Of course, the A is dominant. Or you have B blood, where the B is dominant over, and you can still be a carrier of the O. And then you have AB blood, is when you have type A and B. They can take any blood. They're quite universal. They're quite universal. And then to get type O blood, even though this is the most common blood type, you need 2 of the recessive type O traits in order to be considered type O. So, for example, you can cross blood like you see on this table. You get 50% type A, 25 AB, 25 type B. Then, of course, we have these definitions at the bottom: multifactorial trait, continuous distribution, discontinuous distribution, and linkage groups. You'll have to go back to that and define that later. Next thing we're going to be talking about is just some blood compatibility charts. You'll see it's just a little like a blood donor. Blood type O can only accept O blood types. Type A can receive O or A. Type B can receive B or O, and type AB can take just about everything. Now, it's important to realize, though, that in this situation here, what happens is the only different one is, so AB is universal blood, type O is the least lucky, they can only take type O, but the A blood can take O in itself, the B blood can take O in itself. So everyone can take O. So O is the universal donor, but type AB can take everything, but O can only take itself. So, of course, then there's a table below this. The ABO blood group is just a little chart with some antibodies present. So Group A has antibodies for B, Group B has antibodies for A, Group AB has none, and Group O has antibodies for both, so it can't take any. Of course, antibodies are what fight things, so Group B has antibodies for A because if it gets A blood, it'll just attack it. Of course, the antibody, so Group A, of course, has the A antigen, and Group B has the B antigen because that's what it can accept. And then Group AB has the A and B antigens, and Group O has no antigens at all because it can only accept type O blood. Now, next thing we're going to be talking about is genetics after Mendel. So multifactorial trait. Many times in more complex organisms, traits can show a wide range of phenotypic expressions. Multiple allelism isn't enough to explain this. Therefore, many genes found at many loci are responsible. To note, the plural of locus is loci. Many genes found at many locuses, if you will, are responsible. Many multifactorial traits show a continuous distribution of phenotypes. For example, people in the human population show this type of distribution for all the varying tallness in humans. Traits where only one pair of alleles is involved show discontinuous distribution. For example, tall or dwarf peas, red, pink, or white flowers, or blood types in the ABO system are examples of discontinuous distribution. Continuous distribution is things that vary, like tallness. Everyone's different. It's not like, you can't be a different form of A blood than the next guy. If you're A-type blood, you're A-type blood. You know what I mean? Anyway, next thing is gene linkage. Genes that are located on the same chromosome tend to be inherited together. When genes occur on the same chromosome, we say that gene linkage is in effect. Therefore, we can conclude that humans have 23 linkage groups. These are packages of genes inherited together. The next thing we're going to be talking about is genes, chromosomes, and DNA. Each species of organism has a specific number of chromosomes in each diploid body cell. Humans have a diploid number of 46, and chromosomes are arranged in 23 homologous pairs with one homologue of each pair coming from each parent. So it's like 2 times 23 is 46, and one of those two is, of course, you have mom and dad. So X and X or X and Y. Of course, it's the father who determines whether you are a boy or a girl if he decides to give away his Y chromosome or his X chromosome. Chromosomes can only be seen in cells that are actively dividing. Chromosomes can be photographed and can be sorted into homologous pairs based on their distinctive features. Those are size, shape, type, and banding pattern. The resulting chart is called a karyotype. Karyotypes can be useful in determining whether or not an organism's chromosomes are normal in number and structure. A human karyotype, just like is shown in that picture there, that is a karyotype, shows 22 pairs of autosomes. Autosomes are non-sex chromosomes. Plus at the end, you'll see the XY, it's labeled, is one pair of sex chromosomes. This last pair determines if you are male or female. The female has 2 X chromosomes, so you're always guaranteed to get an X chromosome from your mother. And a male has 1 X and 1 Y that make up this pair. So, your father decides, are you a boy or are you a girl? Sex linkage. Sometimes certain traits are inherited depending on the sex of the individual. The human X chromosome is quite large and contains many genes, while the Y chromosome contains only a few. Genes on the Y chromosome solely determine maleness. Any traits controlled by genes on the X chromosome are called X-linked traits. A gene on the X chromosome in the male has no matching allele on the Y chromosome. So any gene on the X chromosome, whether dominant or recessive, is expressed in males. In females, it is different because there is another gene on the X chromosome to possibly dominate over the trait on the other X chromosome, or vice versa. So, males usually get the short end of the stick because they only have one X pair. So, for example, if a male gets color blindness, which is an X-linked trait, they have color blindness. If a female gets the color blindness allele, they probably won't show it because they have two X copies. Therefore, any faulty gene, color blindness, hemophilia, baldness on the X chromosome is always expressed in males, but rarely in females unless they have two defective copies of the X chromosome. And you will see that's an example. You can see this is a colorblind, you see an example of a colorblind male, and in the Punnett square up top here, you can see that right now, so we have XC, that's an affected girl. So this carrier mother is obviously also colorblind because she carries two copies of the colorblind trait. Oh, my apologies. Sorry. So you can see that this father is a capital C, so he's unaffected. And this mother has a capital C, so she's good, but then her other one's got a little lowercase c. So in this Punnett square here, you can see that he's got so that's an unaffected female, but she's a carrier. You have another unaffected female, but she's a carrier. And then you have an unaffected male, but then that last one there, that poor guy, unlucky guy, is an unlucky colorblind male. A carrier is a person or other organism that has inherited a recessive allele for a genetic trait or mutation, but does not display that trait or show symptoms of the disease. So you can see this colorblindness test, which I utterly fail every single one of them. But you can see that green one, that's like a little benchmark one. That's 12, and then I think that's a little 2 in there. And I don't know, I just see a bunch of jumbles of colors for the other one. Anyhow. Next thing we're going to be talking about is... Oh, there it is. And then, so the next note here, 7.1a, is just a repeat of that note that we literally just looked over. It's just because it's very important, this note about the chromosomes, the DNA, color blindness, everything else. Then the next note is called DNA. All chromosomes are made of deoxyribonucleic acid. You don't need to know that word, you can just call it DNA. So all chromosomes are made of DNA and proteins. In 1944, the structure of DNA was discovered by Watson and Crick. DNA is known as a double helix. It can be described as a twisted ladder. The two sides of the ladder are made up of a repeating sequence of sugar and phosphate groups. The rungs of the ladder are composed of a pair of nitrogenous bases. The building block of DNA is a nucleotide composed of a sugar molecule, a phosphate molecule, and a nitrogenous base. There are 4 types of bases: A, C, G, and T. And of course, T eventually gets replaced by U. The A means adenine. The C is cytosine. The G is guanine. And the T means thymine. These are very important. So we have adenosine, cytosine, guanine, thymine. Adenine, cytosine, guanine, thymine. A, C, G, T. There you go. Of course, you'll see on that chart, that's they have it laid out. They have the phosphate bases. They have the sugar bases. You can see those on either side. And then you have the A always matches with T. T always matches with A. T always matches with A. G only matches with C. C only matches with G. Bases match up with each other is called complementary base pairing. A matches with T and C matches with G, as I said. The code of the DNA is determined by the order of billions of these pairs attached to each other in this helical shape, twisted ladder. I'm going to say that again. The code of the DNA is determined by the order of billions of these base pairs attached to each other in this helical shape, the twisted ladder. A long sequence of these nitrogenous bases makes up a gene, and the order of the bases in this gene determines what product the gene will produce. The sum of the genes in a cell, in turn, determines what kinds of proteins a cell will make. DNA replication. Every cell must duplicate its DNA before it divides. When DNA divides, the bonds holding the middle of the rungs of the ladder break apart, kind of like unzipping, producing two half ladders of the exact same length, or strands of DNA. With these rungs exposed and nothing attached to them, the complementary base is gone. Free-floating nucleotides find their way to this strand and they join with it, completing the new DNA molecule. Note, only A will bind with T and G will bind with C again. That's quite important, if I haven't made that pretty clear. What results is two DNA strands, identical to the other. This method of replicating the hereditary information is termed semi-conservative. So, it's called semi-conservative replication. What happens is, you get the DNA is replicated, so you get a complete copy of DNA, and then the original DNA splits in half, and it pairs with the original DNA. So then you have one original and one new DNA, and then you have conservative is different, where it creates brand new base on its own. So, semi-conservative. This is because the two new DNA strands contain half the original strand. The other half was just created after it was unzipped. That's the way DNA replicates. It's just how it's done. If the original molecule had kept both old chains while completing a new double-stranded molecule was built, the replication would have been conservative. So this is what happens. You'll see in that picture, conservative replication. You can see the original DNA double helix. It stays the same. It doesn't change. And then DNA molecules, after one round of replication, of course, they create their own there, as you can see in the picture. So you'll see semi-conservative breaks in half, and then it makes new ones. And then you'll see that that new red pairs with an original, and then you get two copies, right? Because you need to be constantly replicating the DNA. Next, we're going to talk about transcription and translations. Quite important. DNA never leaves the nucleus. A special molecule called RNA is made in the nucleus, which is a single strand copied from one side of the DNA molecule in the nucleus. This process is called transcription. It then leaves the nucleus to go to the cytoplasm. The difference between DNA and RNA is that RNA is single-stranded, and all the thymine T bases are replaced with the U bases. U means uracil. So now U, uracil, is now binding with A, adenine. So this new RNA leaves the nucleus and finds a ribosome. The RNA is called messenger RNA, or mRNA for short. So you can see there on that DNA and RNA structure, of course you have half. You have thymine, uracil, cytosine, adenine, guanine, of course there, right? And this way, the original master copy of DNA remains safe in the nucleus to make more copies of mRNA, right? So the new mRNA finds its way to ribosomes in the cytoplasm and attaches to them. Now the process of translation takes place after the new mRNA finds its way to ribosomes in the cytoplasm and attaches to them. Translation is when protein is made from the coding found in the mRNA. Proteins are molecules which consist of many different types of amino acids linked to each other from end to end, creating a long chain. There are 20 different amino acids possible in humans, right? So, translation is when protein is made from the coding found in the mRNA. So protein is made from that coding that is found in the mRNA. So protein are molecules which consist of many different types of amino acids linked to each other from end to end, creating a long chain. And there are 20 different amino acids possible in humans. The mRNA sequence, as it is read by the ribosome, and of course most of this you can see in the picture, directs the appropriate type of amino acid to be bonded to the growing protein chain. The mRNA is read to the end until the protein is completely built. So you can see that, right? You can see you got the DNA, cell nucleus, cytoplasm. It finds its way it exits the nucleus, finds its way to the ribosome, and then the ribosome interprets, okay, what protein, what amino acids do I need to be making and putting in my chain here? And then the ribosome does that, right? So therefore, this explains how DNA codes for the mRNA sequence, which in turn codes for the amino acid sequence linked together to form a protein. What makes cells a certain kind of cell is what proteins they make. If all cells contain the same DNA, how can cells be different? Liver, nerve, bone cells, etc. Well, scientists think this is because some cells are able to turn off certain genes to make proteins and turn others off. So, of course, we have cell differentiation. The stem cells, they tell you to make different cells. So, of course, you can make liver cells, intestinal cells, cardiac cells, nerve cells, blood cells, muscle cells, whatever you need. The body just knows, and it says, okay, you need skin cells in this part of the body stat, kind of thing. Discovering how this happens is an area where scientists are working hard to figure it out. This way we can learn how to convert one type of cell into different types of cells. Of course, you might have heard of stem cell therapy. It's mainly what he's talking about there. Next, we have genetic disorders. Any change in a gene that is accompanied by a loss or change in the functioning of the genetic information is termed a mutation. So a mutation is anything that changes the functioning of genetic information. If a gene is altered, in most cases the undesirable information is in the form of a recessive allele. As a result, the harmful effects of the changed gene will not be expressed in the heterozygous condition. In fact, if you have anything in the heterozygous condition, you almost won't even notice. Any factor that can cause a mutation is termed a mutagen. So a mutagen could be UV radiation, X-rays, cigarette smoke, benzoyl peroxide, nitrate and nitrate preservatives, barbecuing, human papillomavirus, HPV, you know what I mean, Helicobacter something or other pylori. Anyway, these are all mutagens. They can mess with you. Be careful. Mutations found in the somatic means body, so any mutations found in the somatic body cells will usually go unnoticed. These cells cannot be passed on to the next generation. A more serious situation would be mutated gametes or sex cells. Here, every cell the offspring grows from will contain this mutation. Also, the mutation can be passed on to many generations. That's not good. Clinical health problems visible at birth are called congenital defects. So again, clinical health problems visible at birth are called congenital defects. Diseases such as hemophilia, albinism, cystic fibrosis, and sickle cell anemia are produced by a single recessive gene mutation. Next thing we're going to talk about is chromosomal abnormalities. The other major type of inherited abnormality involves whole chromosomes rather than single genes. Aneuploidy. Again, aneuploidy is when there is an extra chromosome or the absence of a single chromosome, like Down syndrome. Usually lethal before birth, an extra chromosome is called trisomic. When lacking one of a pair of chromosomes, it's called monosomic. So you'll see on that little chart on the side, trisomy is when, you can see, so when they were ripping apart, accidentally we took an extra one with us, and then we have one less on one side and one more on the other side. Down syndrome is one of the more common abnormalities where the person has 3 copies of chromosome 21. They usually have characteristic faces, eyelids, tongues, and hands, and are developmentally challenged in varying degrees, both physical and mentally. This usually results from non-disjunction, failure of chromosomes to separate or move apart during meiosis. Of course, you can see that again in that chart there. Genetic studies go beyond just at the family level, but studying whole populations, called population genetics. Researchers study the frequency of a gene in the gene pool or the frequency of a genotype in the whole population. Don't forget that. Population genetics and gene pool are highlighted. Pedigrees. This is a type of chart that shows the pattern of transmission of a trait within a given family. These 3 are the most important patterns for simple inheritance. So next I'm going to show you a pedigree chart. And they're the most common patterns for simple inheritance of different things. Of course, you can see on that chart too, you have a male left, female right. They're unaffected. And then affected individuals are, so the square is a man, circle is a woman. If you're affected, you're colored in black. If you're a carrier, heterozygous, then you're half and half. And then deceased, crossed out, sex unspecified, is a little triangle. So the first one is autosomal recessive inheritance. So here, the affected individual must have both defective copies of the recessive gene to be affected. The trait may skip generations, and some can be carriers. Males and females are affected equally. So here you see, right at the top, unaffected father, the mother is a carrier. She has kids. The first son is not affected at all. Second son's a carrier. Then the son marries another unaffected. And then you have a son and then you have a boy who marries a carrier girl, and you have another girl. So then you can see that they have kids, and one of their kids is a carrier. But you can see these crossed lines. That's a little problem. Now, if you'll notice here, it seems that this was some first cousin incest, and two carriers, right? So you have two carriers mated together, which is not good. And then look at this. We've got a lot of problems in this family. We've got this guy is really affected on the corner, this poor sucker. And then we have carrier male, carrier female, so they got lucky. And then one unaffected female, so holy cow, that's not good. And then the next one, so that's autosomal recessive inheritance. And then we have autosomal dominant inheritance. Now this is the bad one. One copy of the affected gene is all you need to be affected. So half of all the children of an affected parent are expected to be affected. It does not skip generations. Males and females are affected in equal numbers. So as you can see here, normal dad marries a not normal mom. They have some lucky kids, but as you can see here, half the kids are affected, and every time it just passes on. Like this guy gets married, has a kid affected, and then they have a kid affected, and then they have a kid affected. So it's not good. Again, that's autosomal dominant inheritance. And X-linked recessive inheritance means that there is no father-to-son transmission because the X chromosome contains the affected gene. It skips generations, mostly in males and quite rare in females. Both their copies of the X chromosome need to be affected in females for it to show. So as you can see, we have two normal parents here. And then we have a daughter who somehow is a carrier of something, marries a guy, and then this poor guy here gets fully affected because he's a male. He's only got one X copy, right? Then it gets passed on to a female. She's a carrier. First one, they have kids. Male is affected. Female's a carrier. Then they have kids. Oh boy. And then another guy's affected. And then we got another carrier here, carrier female. And then bam, she has a kid who's affected. Another carrier female. So then the process is just going to pass along. And the next one here, you got an affected guy. And then you have a carrier who produces two affected people. So sucks for them. And that's X-linked recessive inheritance again. So the next, and I believe, no, not the final thing, but the next thing we're going to talk about is applying genetics. So first is breeding strategies. So plant and animal breeders realize the advantage of controlled or selective breeding. These are highlighted words, controlled breeding and selective breeding, before Mendel manipulated pea plants and figured out all the science behind it. So for example, the earliest farmers quickly realizes the value of saving seeds from the best year's crops. If you save the best year's crop seeds and you replant them, you're going to keep having the best year's crops. The best milk-producing cattle or wool-producing sheep were mated while the livestock of poorer quality were prevented from mating. You can see in this picture, you have a skinny chicken, a bigger chicken, and an even bigger chicken. If you keep mating big chickens, you're going to get a whole lot of big chickens to go with it. You're going to make more money. You're going to have bigger chickens, more meat, more bang for the buck. Once a number of desirable traits have been obtained in a plant or animal, this set of traits can be passed from generation to generation intact as a result of a type of controlled breeding called inbreeding. Now, in plants, inbreeding involves the self-pollination of a desirable trait. In cattle, brother and sister matings are used to maintain a good herd. there's a serious drawback from inbreeding because it's great for a while, but it's only great till it isn't. Now, this drawback is all organisms possess some rare, harmful recessive alleles that don't express themselves in the heterozygous condition. So, on their own, they're fine, but then they start having kids, and then if you keep putting kids with a family member that might also have this recessive allele that you don't notice, all of a sudden you're going to have pretty affected offspring. So, there's a chance that offspring can inherit 2 copies of these harmful alleles. Another drawback of this homogeneity, so that's a highlighted word, homogeneity. Another drawback of this homogeneity is the susceptibility to disease. Another method of selective breeding is hybridization, which selects 2 completely different parents, each homozygous for a different desirable trait. The resulting offspring is heterozygous for many traits and has hybrid vigor because of this variability. I'll say that again. The resulting offspring is heterozygous for many traits and has hybrid vigor because of this variability. So you'll notice, see, you have some, on the picture, you have these hybrids. You have nice big corns. Then you got some little funky looking inbreds over on the side there. Genetic screening is the next thing. This is used to identify those individuals with an increased risk of inheriting a disorder. This is often tied to a group of specific ethnic background because these people share a common genetic background. Screening may involve karyotype studies or biochemical tests. So when I think of this, you think of, indigenous people are more likely to be addicted to alcohol, and they're also a lot more likely to get diabetes because their bodies and their ancestors, they're used to eating such lean diets. And then all of a sudden now, Americans come in and we've got, McDonald's and A&W and all those good things, and they're not used to it, all the sugar and all that's, it's terrible for them. Of course, they're also genetically predisposed to be addicted to alcohol. Genetic counseling is something else. This is offered to many prospective parents and to families with affected children. So this can help educate couples regarding child rearing or childbearing, especially to parents who are, at risk for passing on, some sort of something dangerous to their children, their offspring. So for example, if a parent is at risk and a woman's 35 years old or older, increased Down syndrome risk, has abnormalities in their family, has a child with a genetic disorder, etc. So some people have a genetic predisposition to many diseases such as cancer. If their family has a significant history of cancer, this disease could be inherited. So sometimes you can do something called a prenatal diagnosis. This involves testing of the fetus to check for a problem for which the family is at risk. So as you can see in the picture, just a doctor puts in a little needle and then they take out some amniotic fluid. So the most common technique, as I said, amniocentesis, where a small amount of amniotic fluid is obtained and the genetics can be analyzed. Again, that is amniocentesis. An ultrasound can be used to help position the needle in exactly the right place. It can also detect a large number of physical abnormalities, of course. Everyone knows you got the little ultrasound pictures in your wallet. So, chorionic villus sampling and maternal serum screening, MSS, are also used to test for genetic abnormalities. Again, that is chorionic villus sampling and maternal serum screening. And I believe the final note is applying genetics. So recombinant DNA techniques allow scientists to equip an organism with DNA that is not normally found in that organism. That is recombinant DNA. This new information can cause the cell to produce a specific protein. Simple organisms like bacteria are used because they can reproduce quickly, and the protein can be produced in large quantity. Even human genes can be transferred, so bacteria cells can make human insulins for diabetics. The DNA from the bacteria is called a plasmid, which is circular. It is cut open using a chemical enzyme called a restriction enzyme. They cut at a specific DNA sequence. This enzyme cuts the insulin gene out of human DNA and has the same enzyme opens up the plasmid. So this restriction enzyme is opening up the plasmid, And then the plasmid ends up taking in the human gene and gets closed up again. The DNA strands know where to close because the restriction enzyme cuts every piece with the same DNA codes at the end. They're called sticky ends. So recombinant DNA, complementary sticky ends from different pieces of DNA can be joined together. Recombinant DNA. So you got the vector, the insert gets cut open, bang, we're good to go. So gene therapy. Gene therapy is a scientific technique that is used to help treat diseases by using DNA. The most common type is used to correct hereditary defects. Theoretically, one can insert a proper working copy of a gene into the cells that lack the ability to produce their own protein. Gene augmentation therapy. See, you have a cell with a not functioning gene. You pop in a functioning gene, and then bam, you get the cell functioning normally. Think about insulin. Genes are introduced by splicing this gene into a virus, which can carry the gene to the site in the body that requires the correct copy of the gene. Viruses insert their DNA into the host's DNA. The key here is to get the gene transferred into the stem cells at the correct site. Stem cells are capable of dividing and are not yet producing the product, insulin for example. For the therapy to work, these stem cells must be able to divide, and the new cells must be able to differentiate into specific types of tissue. More work is needed here. There's a whole lot of research going into DNA this, DNA that, just stem cell research, all that crap. Next thing is the Human Genome Project. There's an estimated 20,000 to 25,000 genes in the human genome. That's all of our DNA. Human DNA base sequences, the order of A, C, T, G, have been analyzed, even though there are about 3 million base pairs in our DNA. The results were published in 2003. The order of the bases gives us lots of information. We have discovered that we have the same genes that are found in other organisms, including bacteria. In the near future, scientists hope to develop new treatments for disease by using this genetic information, but we have to fully understand it first. So cloning. There are different levels of cloning. The production of insulin by bacteria is a method of gene cloning because the same new foreign gene is found in all the cells. Growing cells that are identical to each other is called cellular cloning. Producing copies of a whole organism is called nuclear cloning. This is where organisms have the identical DNA in their cells. The first mammal ever cloned in this way was Dolly the sheep. DNA from a sheep was fused with an egg cell, with its DNA removed, of course, and put into a surrogate mother so the new cloned sheep could be born. And you can see those are frickin' identical sheep right there. Holy cow. The ethics of current practices. So this technique and practices discussed here have the potential to alter and direct the path of evolution in many species of organisms, including our own. Changing the genetics of species has the potential for them to cause great harm, like genetically modified organisms, GMOs, food. Human genetic information, according to some, should be private, so one doesn't discriminate or bias on the basis of their genetic makeup. Think about insurance. If you're more or less likely to be a serial killer, I bet your life insurance policy is going to be much worse. Should every human submit their DNA for analysis to help law enforcement around the world? Personally, I don't think so, because I think that this could be used in a lot of bad ways, but anyhow, this is just my opinion. And that is all for the Chapter 6 and 7 notes. I'm assuming this is quite a long video, so good for you for sticking around for 54 minutes, and hopefully this is helpful in studying. Best of luck.